Hi humans! So we'll start with nucleus. Nucleus is found in all eukaryotic cells. It contains the genetic information in the form of DNA. So this is why it controls the cell activities like a boss. So this is the nuclear envelope. So the nucleus is surrounded by a nuclear envelope composed of the outer membrane and also the inner membrane. So the outer membrane is actually continuous to the endoplasmic reticulum, which may be covered with ribosomes for protein synthesis. So the nuclear envelope is perforated by nuclear pore like this, and the nuclear pores allow exchange of substances between the nucleus and the cytoplasm. So what goes in are molecules like protein and nucleotide, which are needed to make ribosomes and DNA, and also hormones for regulation of DNA. And what goes out are messenger RNA and also newly made ribosomes. Chromatin is mainly composed of coils of DNA and also basic proteins called the histones, and they form a bead-like structure, which is also called the nucleosome, and they are regularly packed to form chromatin. During nuclear division, the chromatin condenses to form chromosomes, and during interphase, those remain tightly coiled. Like this, the dark lines, they are called heterochromatin, and those which are loosely coiled are called the euchromatin. And in most animal cells, the heterochromatin is localized near the inner surface of the nuclear envelope. Nucleus is the rounded, darkly stained structure inside the nucleus. It functions to make ribosomes. And the reason it stains intensely is because of the large amounts of DNA and RNA it contains. So that's it about nucleus.
Plasma membrane. Plasma membrane is a semi-permeable membrane. It is made up of proteins and lipids. The main composition of plasma membrane would be the phospholipid bilayer. It consists of hydrophobic tail and a hydrophilic head. So the hydrophobic tail is made up of two fatty acid chains, normally carbon 16 and carbon 18 fatty acid, while the hydrophilic head is actually made up of polar phosphate group. Thus, this makes this plasma membrane amphipathic as it has both the hydrophobic and hydrophilic regions. Cholesterol is actually embedded in between the plasma membrane or the phospholipid bilayer as it strengthens the bilayer by increasing its fluidity but at the same time decreasing its permeability. So another major component of this phospholipid bilayer would be the proteins, which are involved in transport of macromolecules from the external environment into the cell and from the inside of the cell to the external environment. So one of the proteins would actually be pore protein. This pore protein is involved in passive transport, which does not require energy, while carrier protein is involved in active transport, which requires energy, and it also has binding sites for the macromolecules to bind themselves before they get transported across the plasma membrane. This phospholipid bilayer, as you saw earlier, is quite thin. Its actual thickness is only 5 nanometers. This phospholipid bilayer is actually also known as a fluid mosaic model. It is known as fluid mainly because the components of this bilayer, such as the lipids and proteins as we saw earlier, are not exactly static. Instead, they are always moving laterally or sideways throughout the membrane, and it looks as though they are dancing. They are also called mosaic because the plasma membrane or the phospholipid bilayer is composed of many different kinds of macromolecules such as transport proteins, glycoproteins, glycolipids, and also cholesterol. So, this plasma membrane is said to have a dynamic structure. We just saw the structure of a phospholipid bilayer, so now let's look at the functions. So, mainly it acts as a barrier separating the content of the cell from its outside environment. Besides that, it also regulates what enters and exits the cell by only allowing selected substances into the cell. The cell membrane is primarily composed of lipids and proteins. Lipids help maintain flexibility, and proteins assist in transfer of molecules across the plasma membrane. Besides all this, the plasma membrane is also involved in maintaining homeostasis by diffusion and osmosis. Last but not least, it serves as a base of attachment for cytoskeleton in some organelles and cell walls, whereby a network of fiber on the cytoplasm will support the cells and maintain shape of the cell. So that's about it. Something wrong with the video. The video.
It's okay, just leave it there.
Shit, damn it! I didn't stop. I just spoke without even stopping.
How to this one? Huh? How to stop now? How to stop?
This is a Golgi apparatus. Golgi apparatus is located within the cytoplasm near the endoplasmic reticulum and nucleus. Golgi apparatus can be found in all eukaryotic cells. Animal cells have less and larger Golgi apparatus, while plant cell has more and smaller Golgi apparatus. Golgi apparatus consists of stacks of flattened membrane-bound sacs called cisternae, together with a system associated with vesicles. Forming phase is where protein is packaged and modified, while maturing phase is where vesicles containing the modified product are pinched off. The functions of Golgi apparatus is chemically modifying, packaging, and transporting of materials. Protein, carbohydrate, phospholipid, and other molecules are biochemically modified in Golgi apparatus. This is a process of producing protein. Next is producing of secretory cells. Secretory cells is storage of materials which is respond to environment signals. Followed by excretion of carbohydrates and formation of lysosomes.
What's up? In this video, I'm going to show you an explanation on the organelle central. So let's see. Okay, let's see. Let's represent this pen as a central. Okay, central is a hollow tube. It will be something look like that. Okay, central is 0.5 millimeter in length. It is 0.2 millimeter in the diameter. So, in the nucleus, if two central were to meet, they will lie something like that. They will be right angle to each other. So let's represent this as another central and this as a central. This will be roughly the position it will be like. During cell division, central will be positioned like that, and they will form something like a jelly, a net-like structure called the spindle fiber. So this spindle fiber aids in the cell division in mitosis and meiosis. They will divide the cell into two. So in short, we can call centrioles as the organizers of spindle fiber. Since central is involved in the formation of microtubules, this makes central important for the production of cytoskeleton in the cell, which is the structure of the cell. Okay, let's imagine that this is a cilia. Okay, and this is a cytoplasm. So the basal body is always embedded in the cytoplasm. Okay, cilia lining in the respiratory tract of mammals will move the mucus towards the throat. Let's imagine that this is the cilia lining, it will move the mucus to the throat. Something like that. So in the same way, the cilia lining in the oviduct will move the secondary oocyte towards the uterus. Let's imagine that this is the cilia lining in the oviduct. The cilia will move, the secondary oocyte will be moved towards the uterus. So that's about it, the function of cilia.
In my head, this is where we all came from. The dreams we have, the love we share. This is why we're waiting for. And in my
Hey guys, what I did just now needs energy. And where do energy come from? So what is mitochondria? Okay, you see, mitochondria is an organelle found in our body. Mitochondria is rod shaped, it is small, and it is 1 to 10 micrometer and mitochondria have two surfaces, the inner and the outer. The outer control the entry and exit of materials from the mitochondria, while the inner is a membrane which is folded. And okay, you see, cristae are shelf-like extension within the mitochondria. It has a large surface area and it's for attachment of stored particles. And next, the matrix. The matrix is the remainder in the mitochondria. It contains protein, lipid, and DNA. Mitochondria have three functions. The first function, of course, we know is the synthesis of ATP and energy. The second is oxidation phosphorylation. We never heard about this, but this is my question. What is oxidation phosphorylation? Yeah, this is something new and we should discuss it later. The third function is the sites of Krebs cycle. Krebs cycle is the enzyme involved to make ATP and energy. So this is the fundamentals of the energy production in the mitochondria. Okay, mitochondria found abundance in the liver and muscle cell. You see, as mitochondria increases, surface area increases, which increases the number of cristae, so more energy can be used for the body. So thank you, that's about it. Peace.
Cells are surrounded by a relatively rigid wall which is secreted by the protoplast called the cell wall. So there's primary cell wall and also secondary cell wall. The primary cell wall is actually laid down during cell division and it consists of a cellulose fiber running through a matrix of polysaccharides, whereas the secondary cell wall is actually the thickened primary cell wall. So this is one plant cell and this is another plant cell. The middle lamella acts as a glue to hold the neighboring cells together. There are also mini pores in the cell wall to allow plasmodesmata to pass through, forming living connections between the cells. So the communication and coordination between the plant cells become easier because the molecules and ions do not have to pass through the cell surface membrane. They can just pass through these pores to the other cells. Lignification. Some cells like silent vessels or sclerenchyma undergo extensive lignification. So this is whereby lignin, a complex polymer, is deposited in all cellulose layers. Lignin cements and anchors cellulose fibers together, give extra tensile and compressional strength. So the functions of the cell wall, first one, as I said before, support and strength, SS. And next, turgidity, because as you can see here, the cell wall is very rigid. So when water enters by osmosis, the cell is resistant to expansion. So the cells will not burst when exposed to any dilute solution. However, it becomes turgid. Shape, not the body shape, but the shape of the cell. The ability of cells to stretch is actually determined by the arrangement of the cellulose fibrils. So the cell wall will help to control the cell growth and also the shape of the cell. Lastly is the cell walls actually develop waxy cuticle on the exposed epidermal surface. So this can actually help to reduce water loss and risk of infection in a plant. So that's it about cell wall. Bye.
So now we're going to move on with chloroplasts. Photosynthesis occurs in eukaryotic cell structures called chloroplasts. Chloroplasts are basically large organelles that contain a green pigment called as chlorophyll, which traps light energy so that photosynthesis can take place. They are only found in plant cells and some protists. Within the chloroplasts are small grains or grana that absorbs light energy for photosynthesis. Thylakoids are small, flattened sac-like structures that are often stacked on top of each other in the grana. They serve as the sites of conversion of light energy to chemical energy. Each chloroplast contains about 40 to 80 grana, and each grana comprises about 5 to 30 thylakoids. The thylakoids are surrounded by the stroma, which is a dense fluid. This is the site where reactions occur and also where carbon dioxide is converted to carbohydrates. Two or more grana are connected to each other by intergranal lamella. In this way, the lamella acts as a skeleton of the chloroplast, maintaining efficient distances between the grana, thereby maximizing the overall efficiency of the chloroplast. Each chloroplast is also surrounded by a double-layered membrane, that is, it is enclosed by two membranes separated by an intermembrane space, and these two double-layered membranes are known as the chloroplast envelope. The membranes act as a protective covering and keep the chloroplast structures enclosed. Starch exists in the chloroplast in the form of tiny lumps called granules or grains. These are present because they are the insoluble storage carbohydrate product of photosynthesis. Chloroplasts also contain the smaller type of ribosomes, that is the 70 Swedberg ribosomes, which is the same type as those freely distributed around the cytoplasm of prokaryotic cells. Each chloroplast also contains one or more molecules of small circular DNA. Chloroplast DNA contains many of the genes necessary for proper chloroplast functioning. It also contains lipid droplets called plastoglobules. Plastoglobules are studded with enzymes which are involved in thylakoid lipid metabolism. The process of photosynthesis is divided into two reactions: light-dependent reactions and light-independent reactions. First, I'm going to focus on the light-dependent reactions. It occurs in the granum of the chloroplast. Energy from the sunlight is absorbed by chlorophyll to split the water molecules into hydroxyl ions and hydrogen ions. This process is called as photolysis of water. Each hydroxyl ion is neutralized by releasing its electrons to the chlorophyll. A neutral hydroxyl group is then formed. Four hydroxyl groups combine together to form water and oxygen. Oxygen then leaves as the waste product. Each hydrogen ion receives an electron from the chlorophyll and is neutralized. A neutral hydrogen atom is formed. The hydrogen atoms are used in light-independent reactions to reduce carbon dioxide. Now I'm going to focus on the light-independent reactions. It occurs in the stroma of the chloroplast. Hydrogen atoms produced during light reactions are used in light-independent reactions to reduce carbon dioxide to basic units of glucose. This reduction of carbon dioxide process involves a series of complex chemical reactions which requires enzymes. 6 units of CH2O combine together to form a molecule of glucose. Glucose is then converted into starch, cellulose, other sugars, and lipids. When combined with nitrogen, glucose is converted to a protein.